Okay, so today's lecture is about tumor immunology and the immune mechanisms that go about to monitor the development of tumor cells and to prevent the establishment of tumors. So the learning objectives of this lecture is to highlight the interactions between the immune system and malignant cells, to talk about the effector cells that are instrumental in killing off tumor cells before they start to grow into a solid or a systemic tumor. But then, just like you've learned in microbiology, you have this constant evolutionary battle between the immune system and pathogens like the microorganisms or tumor cells to try to evade the immune mechanisms. And then after that, I'll talk about the nature of these antigens expressed on tumor cells that make them immunogenic. But the problem here is that because these are tumor cells, they are very similar to normal host cells. And so this is what the immune system has a problem of discriminating between what is a tumor cell and what is a normal antigen, self-antigen. And then finally, I will talk about the current breakthrough we've had in manipulating the immune system to make it more effective in the treatment of cancer. And in this respect, it's met with a lot of success in the past 15 years through novel genetic engineering techniques. So, first of all, in the early 20th century, before immunology was even understood, it had always been suspected that there was something in the body which protected us from the growth of cancer, the development of cancer. And in the 50s and 60s, immunologists created a model whereby they could induce tumors in laboratory mice by injecting them with various carcinogens, various chemicals. And with this ability to establish tumors in mice, what they then did was to perform experiments like this, where they would inject irradiated tumor cells into a mouse. Now, why would we irradiate the cells? What would that do? Sorry? So basically, by irradiating cells, giving cells a sublethal dose of high energy X-rays or ionizing radiation, what you're doing is disrupting the DNA of the cells, but not such high doses that you can. So in other words, you are preventing the cells from dividing, but they're staying viable. And this is a common practice in these type of experiments. So essentially what you are doing then is injecting these mice with inactivated tumor cells that aren't able to divide into a solid tumor, but they're acting as targets. So you inject the mice with these target cells, and after a few weeks, you inject then the same tumor cells, but that haven't been irradiated. So it means that these cells will be able to proliferate and establish a tumor. But what you find is that no tumor develops. If you just injected live tumor cells into the mice, you would get a solid tumor developing like this. So it suggests that by injecting these irradiated tumor cells, you're inducing an immune response such that when you get a secondary challenge with live viable tumor cells, that immune system's able to kill off those cells before they can grow. And from these experiments, it was also established that this immune response was specific for that initial tumor, those initial tumor antigens only. Because if they injected a different kind of tumor into those animals in terms of a secondary challenge, then that second tumor injection would develop into tumor mass. So this was showing that there was immune specificity involved. And if before we really understood particularly solid tumors, how they developed, why they developed, pathologists would look at biopsies of tumor masses and find that tumors were surrounded by huge infiltrates of monocytes, T cells, natural killer cells. When you go into the clinic suspected of having a tumor, one of the first things the clinician will do is check your lymph nodes to see if there's any swelling, which could indicate that you could start of having metastasis, or there is immune activity going on in the draining lymph nodes to deal with that localized tumor. From experiments like the one I just showed, it was shown that T cells reactive to tumors obviously could be adoptively transferred. Subsequent experiments in mice and also in children who are born with the immune deficiencies indicate that cytotoxic T cells and natural killer cells have a very important role in the so-called tumor surveillance, that they are essential in killing off tumor cells. But even though we know that there is this immune surveillance mechanism, people still get tumors. And why is this the case? So as a result, through years of studying cancer, we've now modified this idea of the immune surveillance against tumors to define a dynamic relationship between tumor surveillance and tumor evolution to escape tumor surveillance. And this revised hypothesis of tumor surveillance is that you first get in a normal body, through our lifetimes, every proliferation or regeneration of tissue, there are mistakes in the DNA which are potentially mutagenic and can induce uncontrolled growth. But as soon as those modified cells start to appear, they're immediately recognized by cytotoxic T cells or NK cells and eliminated before these unregulated growth cells can establish themselves. Over time, you will get variants or single cell clones that can evade these immune mechanisms. And so you get a steady state balance between immune cell killing and survival of tumor cells. And over time, these surviving tumor cells will adapt to create mechanisms to undermine that immune response. And at this point, then the tumor cells win and they establish and they grow to such a big size that the immune system can't handle them anymore. And then they can also break through into the endothelium or the lymphatics to then metastasize and occupy other sites of the body. Now, the evidence for this hypothesis comes from two observations. The one which is relatively recent, about 15 years ago, is that there was a woman who developed a melanoma, which is particularly notorious for being metastatic cancer. And after years of chemotherapy, aggressive chemotherapy, she was seemingly cured of melanoma. And she lived for another five years or so, free of cancer. But then she died, and her kidneys were donated to two patients. After 2 or 3 years, these patients developed melanoma, the same melanoma that she suffered from. The conclusion from this was that despite that chemotherapy, even though it seemed like she'd been cured, there were pockets of melanoma cells still living within the kidneys, such that once they were transplanted, these recipients were under immunosuppressive drugs. These melanoma cells were allowed to break out. The other observation is that a relatively common occurrence in people who are receiving immunosuppressive therapy develop B-cell lymphomas. These are mainly due to this herpes virus, Epstein-Barr virus, which can infect normal B cells and then modify them into what we call immortalized cells. In other words, they can change the DNA to prevent these B cells from dying through old age and just to continue growing, as a result of which these people suffer from these lymphomas, B-cell lymphomas. And this is called post-transplant lymphoproliferative disorder. Another observation is that with modern genetic engineering, we were able to knock out particular genes in mice and then look at the phenotype. So in these experiments, if you knock out perforin, which if you recall is one of the cytotoxic granules that cytotoxic T cells and NK cells need to kill their targets. So basically there's no NK and CTL activity in these mice. These mice often develop lymphomas. So this gives an indication of the importance of the role of this arm of the immune response in dealing with systemic tumors. If we knock out RAG, which remember creates an active functional B cell or a T cell receptor, or STAT1, which is important signal transduction messenger for cytokine receptors. So essentially we're creating a severe combined immunodeficiency. There's no functional B or T cell activity in these animals. You find that these mice develop tumors in the gut, and also breast tumors. So it suggests that the immune system is very important in monitoring mucosal tissue for unregulated growth. And then this other subset of T cells, gamma T, delta T cells, we knock that out, we find that these cells are an important source of the type 1, type 2 interferons, and these mice develop skin tumors. So again, these gamma T cells play a role in immune surveillance within the epithelial tissue of the skin. Now, what about the evasion mechanisms of the tumor? So first of all, like I said, because these are minor modifications of self-proteins, their antigenicity is often very similar to normal self-proteins. So the immune system has to recognize these modifications compared to normals. And many times you are killing normal host cells, even if you are trying to target the tumor cells through cross-reaction. The other thing is that over time, as these tumor cells develop, they can manipulate these co-stimulatory molecules like CTLA-4. Remember, CD28 on T cells needs to see B7 on dendritic cells. The downregulatory signaling molecule is CTLA-4. Many of these tumor cells can induce CTLA-4 in the T cells attacking them, so blocking their activity. Another thing tumor cells can do is promote the growth of regulatory T cells, and so again block cell activity against them. They can also upregulate co-receptors like PD1 ligand. PD1 ligand acts similarly to the TNF receptor system. So when these tumor cells are expressing PD1 and they're attacking cytotoxic T cells are expressing PD1 ligand, as soon as those cytotoxic T cells try to kill the tumor cells, the cytotoxic T cells themselves are killed. Then, because these developing tumors are highly mutagenic, they're constantly changing their antigenic epitopes. So every time you have cytotoxic T cells that are recognizing antigenic epitopes on class 1 MHC, with the new proliferation of tumor cells, they could have changed their antigenic epitopes so they're no longer recognized by the cytotoxic T cells. And then finally, in terms of solid tumor masses, as they grow into a mass, what they can also develop is a solid, a thick collagen barrier around the tumor mass that prevents things like antibodies or cellular immune cells breaking through that barrier to get into the underlying tissue to deal with it. Now, what about the antigens recognized by the immune system that are specific to tumors? And we divide them into two major groups. One which are called tumor-specific antigens, and the other that are called tumor-associated antigens. The difference lies in the language: specific, various, associated. Specific refers to antigens which are only present in these tumor cells. They are not present in normal host cells. And so they have come about as a result of mutations in the genes coding for those proteins. And as a result of that, as a result of those mutations then confer within those proteins different functions. I think probably Dr. Barnes already talked to you about this. Many of these mutations relate to proteins involved in signal transduction, in cyclin genes involved with cell replication, cell survival, and so on. We also call these oncogenes. Many of these mutations can be induced by retroviruses. These retroviruses, which can insert themselves, which are initially RNA viruses, they can insert themselves into DNA. I'll talk about that later on this week when I start about the virology lectures. So as a result of these mutations, they create what are called neoepitopes, novel epitopes different from self-peptides, which are tumor specific. Now the problem is that we only know about these neoepitopes mainly through experimental studies by inducing tumors in mice. It's very difficult to discover neoepitopes in humans because as soon as tumor cells arise expressing these neoepitopes, they're generally killed by the immune system. So it's only in the case where an actual tumor is developed that we can actually isolate these things and see what are these antigenic sites recognized by anti-tumor cells. The other class of tumor antigens are these so-called tumor-associated antigens. These are normal self-proteins. But the thing is that in the development of the tumor, where gene regulation has been completely gone overboard, there are many genes which, as a result of development, as we go into adulthood or as we mature, many of these genes are switched off. But because of the tumor mutation events going on within the genetic program of these cells, many of those tightly regulated genes aren't controlled anymore and they proliferate. Examples of these have you know about alpha-fetoprotein? So alpha-fetoprotein is a protein which is involved in embryonic development. As the embryo gets to, after a few months or years, that alpha-fetoprotein is switched off. But in liver cancer, you get high levels of alpha-fetoprotein circulating in the blood. So it's a common biomarker for liver cancer. And the same for the so-called melanoma-associated antigens, or MAGE. So these are normal proteins which are only expressed in one part of the body, the testis. But in melanoma cells, somehow the regulation of these organ-specific genes are broken down, and then these melanoma cells express these genes. They're normal proteins, but they're inappropriately expressed. Another example is for breast cancer. There's HER2/neu, which is the epidermal growth factor receptor, COB2, expressed on breast tumor cells, And because they're overexpressing these receptors, it means that they're highly induced by epidermal growth factor to proliferate. Again, they are normal proteins, but they are overexpressed in this case, allowing for this growth advantage of the breast tumor cells. Other types of normal host proteins which are modified is not so much in terms of the amino acid sequence, but in terms of post-translational modification like glycosylation. And this is in the case of this mucin 1 protein, where reduction of glycosylation confers to that tumor-associated protein a different physical characteristic. Going back to these tumor-specific antigens, as I said, many of these antigens are virus-specific or virus-induced. An example of this is the human papillomavirus, which there's certain strains, 16 to 18, where as part of the virus structure, there are various proteins which, when getting into a cell, can induce a cell to become oncogenic. And so one of the biggest success stories in the past 20 years was the development of the papillomavirus vaccine, which has significantly brought down the incidence of cervical cancer in women. The same goes for the development of hepatitis B virus vaccine. This, it's not so much an oncogene in this case with hepatitis B, but it's the result of what you think of as a type 3, type 2, type 4 hypersensitivity reaction. I don't know if I talked about this before, but the thing about hepatitis B and hepatitis C is that they are strongly associated with liver cancer. People who get these viruses will have a high chance of getting liver cancer 30 or 40 years after they were first infected. The way the cancer develops is not due to the virus itself, but due to the immune system attacking the virus-infected hepatocytes. Because these viruses don't kill the cells that they infect very much. But what they do is induce the expression of virus-specific proteins on the class 2, class 1 MHC of the infected hepatocytes. So when they start replicating and the virus antigens are expressed on these cells, the immune system kills those infected hepatocytes, and then the liver cells need to start growing again to replace the ones that have been killed. The virus will start replicating again, get killed by the immune response, and you get another wave of replication. Over 30 or 40 years, there's so many waves or rounds of hepatocyte replication that's gone on that these hepatocytes start to accumulate mutations in the genes, allowing them to develop into a liver cancer. So then the development of the hepatitis B vaccine has been very significant in dropping or reducing the frequency of liver cancers. Because another feature of hepatitis B and hepatitis C is the ability of these virus infections to become chronic. Another type of relatively frequent tumor, occurs with B cells, B cell lymphomas. I think Dr. Rehan might have talked about this in the pathology. But if you recall, when I talked about the development of B cells through that VDJ rearrangement region through that enzyme TDT, that enzyme can also create mutations within genes involved in the cell cycling function in B cells. So when that goes wrong, you then get the outgrowth of a B cell lymphoma. So this is one of the causes. So now let's talk about the new advances and developments and success stories in immunotherapy. There are two major successes. The one is through the development of what is called a chimeric antigen receptor, or CAR. Before we talk about CARs, first of all, someone tell me what is the major difference between the way an antibody and a T cell receptor recognizes antigen? What is the major difference between the way an antibody and a T cell recognizes an antigen? They may be the same protein. Right, so the antibody recognizes the whole structure, the whole protein. The T cell is limited because it has to recognize that epitope on a particular MHC molecule, right? So basically what the chimeric antigen receptor involves is taking out the gene which codes for the antigen binding region, the CDR regions of an antibody molecule. Now let's say this is specific for a particular tumor antigen, or in this case, for markers which are expressed on B lymphocytes, CD20, right? So you take out the gene sequence which codes for this antigen binding region, and then you link it to the signaling sequence of a T cell receptor. So now then you insert that into a functional cytotoxic T cell. So basically what you have then is a cytotoxic T cell with a B cell receptor that can recognize antigen without being limited by MHC restriction.
So they can directly attack
They can directly attack any surface target and signal to that cytotoxic T cell to kill, just like a CTL does, kill that target cell without needing MHC restriction. So it means that by using or by modifying cytotoxic T cells of anybody, you can create this effector cell. If it was limited by MHC restriction, it meant that just like tissue transplantation, you could only do this for people of particular MHC phenotypes. It's not as simple as that because I don't know if you're following the news lately about these two COVID-19 vaccines. The way you do this is to introduce, once you've got the gene sequence coding and made this hybrid gene containing the antibody recognition and the T cell receptor signaling, you create the messenger RNA which codes for this protein. And you insert that messenger RNA into a vector, like a retrovirus, which you then need to infect a cytotoxic T cell, such that this vector will transcribe and have that been translated to give you this functional receptor. So it's relatively simple in a molecular biology lab, but it's still not as straightforward as what's described here. But it has been used in clinical trials and has very high success rate, particularly against lymphomas where we're developing cytotoxic T cells against these CD20 expressing B cells. Another mechanism of immunotherapy involves expanding tumor-specific cytotoxic T cells in vitro. So basically what you do is you take blood out of this patient who's got cancer, knowing that within that blood there is a certain proportion of cytotoxic T cells against the cancer. So then by treating it with cytokines like T cell growth factor or IL-2, you can then proliferate those cytotoxic T cells in the test tube and then infuse that back into the patient. That has some success, but another mechanism that I was involved in a clinical trial with was where this was in the case of someone with liver cancer related to hepatitis C, is that you do a resection of the tumor, and then in the laboratory you mix up those liver tumor cells with blood that you've taken from the patient. The idea there is that you are presenting tumor antigens from the liver cancer to the person's dendritic cells and macrophages. So you're feeding the antigen presenting cells with the tumor antigens in vitro. And then you inject that back into the patient. So then these tumor antigen presenting APCs are presenting them to the CTLs, expanding them to then get an immune response. We now use a lot of monoclonal antibodies directed against, for instance, for breast cancer, this HER2-neu, the EGF receptor. So by using monoclonal antibodies to block the EGF receptor on the breast cancer cells, you're preventing them from receiving the growth signals from EGF. The problem with this is that monoclonal antibody therapy, those monoclonal antibodies have a relatively short half-life. So you have to keep on repeating the infusions. The other major breakthrough is this process called checkpoint blockade. So if, as I said, remember these two polarized co-stimulatory molecules. CD28, the activation signal, binds to B7 on the antigen presenting cell with a target. The CTLA4 on the same cell recognizes the same ligand B7, and that instead gives a negative blocking signal to that cytotoxic T cell, right? And tumor cells can stimulate the cytotoxic T cells attacking them. They can stimulate these cells to express CTLA4 and not CD28. So in other words, they can downregulate these attacking cytotoxic T cells. So with checkpoint therapy, what you can do is to inject monoclonal antibodies against CTLA4. These monoclonal antibodies will then block those negative regulatory signals on those CTLs so that they're only getting CD28 against the B7s on the cytotoxic, on the tumor cells. And so they'll be super activated to kill them. And this has had quite a lot of success. Then there are other types of experiments of weaponizing antibodies. So basically, what you can do is to make monoclonal antibodies against various tumor-specific antigens and then tag on the FC region various poisons or radioactive substances, such that when these antibodies bind to these tumor cells and then get internalized, those poisons will kill those tumor cells. And this has some promise, but there's still some problems associated with this mechanism. But still, well, another relatively new discovery is the role of the type 1 interferons as adjuvants in tumor therapy. Now, I've talked about type 1 interferons as being predominantly antiviral, interferon alpha and interferon beta. But it's coming to light in the past 10 years or so that these type 1 interferons also play a very important role in stimulating the immune system. They're not just antiviral. And one of their most significant roles is in terms of helping the conventional anti-cancer treatments like chemotherapy and radiation. So in other words, when these patients that are receiving these therapies are also infused with type 1 interferons, there is better prognosis in terms of reducing the tumor load. So somehow, in addition to these physical treatments of radiation and chemotherapy, the addition of interferon to boost the anti-cancer immune system also helps to promote this cancer-killing effect. And the role of this type 1 interferon is quite multifunctional in terms of activating dendritic cells to present tumor antigens to the CTLs, to increase CTL and NK cell killing, decrease the activity of regulatory T cells, activate M1 macrophages, and so on. So, as I said, we now know, and there's constant research groups trying to identify new tumor-specific and tumor-associated antigens to try to develop cancer vaccines against these to prevent the outgrowth of tumors. The problems with this well, as I said, the success stories have been with two particular viruses, hepatitis B and the human papillomavirus. One of the problems with with trying to develop a vaccine, and I'll talk about this in a later lecture about vaccine development, is that many proteins aren't necessarily immunogenic. If you just by purifying a protein and injecting it into a person thinking that you can develop an antibody response, it won't happen. What you need to do is stimulate all arms of the immune response, the antigen presenting cells, the T cell, T helper cells, and as well as the B cells. So even though you can isolate tumor-specific antigens as a potential vaccine, what you need to do is try to boost all arms to get a good B cell memory response, as well as stimulating cell-mediated immunity. Then the other thing you also need to do is to try to combat or get past these immuno-blocking mechanisms of the tumor cells. And you can do this by checkpoint therapy or whatever. Okay, that's it. Thank you.
I think someone wanted you to repeat TDT.
So someone wanted me to repeat what I said about TDT. What this referred to was the role of this TDT enzyme in the development of B-cell lymphomas. Because the function of TDT, if you remember, is in the development of the B-cell and the T-cell receptor in terms of increasing the variability of the CDR hypervariable region in the antigen binding site. And TDT does this by inserting or deleting randomly nucleotides to the genetic code coding for that sequence. The trouble is that TDT can also do this insertion and deletion within particular genes associated with cell proliferation, B cell proliferation. So when it does this, it makes that B cell become malignant.
The retrovirus is only selected for cytotoxic lymphocytic antibodies.
So the question, that's a good question, is the retrovirus only selective for CTLs or can it infect any cells? No, it can infect any cells because essentially you're just doing it blind. But the thing is that even though you might get another parenchymal cell, an epithelial cell expressing this chimeric antigen receptor, it's not going to function. So it's only functioning within the CTLs. What is the factor EGF? It's epithelial growth factor, which is obviously a growth factor for epithelial cells. Okay?
Doctor, do you have any tips for SAQ immunology?
Do I have any tips for the SAQ immunology questions? No, because the SAQ hasn't been set up yet, so I have nothing to say. Okay. All right. Thank you.